All right, this is going to be the first of several, hopefully short videos that will talk about chemoorganotrophic catabolism in bacteria. So you guys will remember from before that catabolism is breaking down things, breaking down chemicals in order to release energy and electrons for further use. And so generally the electrons are going to be captured and held by the NADH and the FADH2. And then these full electron carriers will take electrons to the electron transport chain, and it is primarily in the electron transport chain, at the end of the electron transport chain, that ATP will be generated through oxidative phosphorylation. In any case, we'll get back to all of this here in just a few pages, but just as a refresher, catabolism is breaking down chemicals to release usable energy and electrons. So you guys will remember from a previous time that we talked about the metabolic or nutritional classification names, where the first part is where the cell gets its energy. So the options for that are chemo or photo, if it gets energy from chemicals or energy from light. The second part is where the cell gets its electrons. So the options for that are organo if the electrons are from organic compounds, or inorganic compounds, or litho if the electrons are from inorganic compounds. What is not represented here at all is the third part of the word, is where the bacterium normally gets its carbons, whether it is an autotroph that fixes carbon dioxide into organic carbon, or whether it just eats organic carbon from the scratch. But it's just not in this. So in terms of chemoorganotrophs, these will actually be chemoorganoheterotrophs, because they will be getting energy, electrons, and carbons, all from organic compounds. We have three options to do that, and that is what is going to be covered in this set of videos. The first option is aerobic respiration, and this is when the electrons go to the inorganic electron acceptor, oxygen. Anaerobic is when the electrons go to inorganic, not oxygen. And fermentation is when the electrons go to an organic compound, because they all start with glucose, and so the differentiation is where the electrons end up once you have liberated them from the glucose. Where do they go from there? And then, if you guys will remember back to that reduction potential chart, the eventual ending place of the electrons indicates how much energy is released. We will, in a different day in class, get to chemolithotrophy and phototrophy. So this, I'm sorry about the glare. This was the best I could do for the glare in the house. All right, this is a figure from your book, Chemoorganotrophic Fueling Processes. So again, we are starting with an organic compound to get our energy and electrons. So if things are respiratory processes, then they have an electron transport chain. And so hopefully you remember that electron transport chains have to be in membranes. In the bacterial cell, this has to be the plasma membrane, because that's the only one that it has got. So both anaerobic respiration and aerobic respiration have electron transport chains. The biggest difference in them is the final electron acceptor. So both types of respiration use inorganic electron acceptors. This is a big difference from fermentation. So aerobic respiration always uses oxygen as the final electron acceptor and turns it into water. So this will be the respiration that you guys are most familiar with from Gen Bio. Bacteria can also do anaerobic respiration, which has an electron transport chain with external inorganic electron acceptors, but these are not oxygen. They're going to be things like sulfate or nitrate or carbon dioxide, but they are going to be external to the rest of the processes of the cell. and anaerobic respiration also has an electron transport chain. Because of the electron transport chain, ATP is made by oxphos, oxidative phosphorylation. We will also make a tiny bit of ATP by substrate level phosphorylation. That is what happens in glycolysis and the Krebs cycle, but it's just a tiny, tiny amount actually. So the majority of ATP is made through oxidative phosphorylation, which is the ATP synthase powered by PMF that is typically drawn at the end of the electron transport chain. Our other option as bacteria, if we were pretend that you're a bacterium, for getting energy and electrons out of organic compounds is fermentation. So fermentation is distinguished from either of the respiratory processes because it only happens in the cytoplasm. It does not have an electron transport chain component, and it uses something called endogenous or internal electron acceptors. And so these will be compounds that are already part of the glycolytic process. So glycolysis makes pyruvate. If pyruvate, part of glycolysis, is then used to accept electrons, that makes it an endogenous or internal electron acceptor. And so it is these, can you guys see that? Yes, internal electron acceptor. That sets fermentation apart, as well as the fact that it occurs in the cytoplasm. Fermentation only uses substrate level phosphorylation, which, if you guys will remember, is the passing of a high energy phosphate from one compound to another, but does not use ATP synthase and happens in the cytoplasm and not in the membrane of the cell. So if you are doing chemoorganotrophic catabolism, these are your three options.